All right, Guru Nation, this is a very special video. I'm going to actually do separate videos on each of these companies that we're going to cover because it's super important. But those that know me know that I'm really interested in CRISPR technology, gene editing, and I've been obsessed with Jennifer Doudna, and she actually wrote this book, A Crack in Creation. But she's actually the subject of a book that I've been reading called The Code Breaker, written by Walter Isaacson. So we actually wrote an article on her in Latinos in Clinical Research. So this is the actual book, The Code Breaker. I recommend this book. I'm listening to it for the second time. So if you want to know in full details the story of Jennifer Doudna as the kind of the central character with a bunch of side characters, I'm going to just introduce some of them to you in this video. And like I said, each of these people actually deserve, and companies that they ended up founding, deserves their own video. So let's go through this article really quick. Berkeley biochemist and Nobel laureate created CRISPR technology. So in 2020, University of California, Berkeley biochemist Jennifer Doudna won the 2020 Nobel Prize in Chemistry, along with colleague Emmanuel Charpentier for the co-development of the CRISPR-Cas9 complex, which is a genome editing breakthrough that has great significance in biomedicine. Doudna and Charpentier, director of the Max Planck Institute for Infection Biology, shared the 10 million Swedish krona, more than $1 million prize. So this is Emmanuel Charpentier. We're going to get to her in a little bit. She's equally as interesting as Jennifer Doudna. In the spring of 2012, Doudna and her collaborators turned the curiosity of nature into an easy-to-use tool that can edit DNA. CRISPR-Cas9 enables scientists to rewrite DNA, the code of life in any organism, including human cells. There's actually trials going on right now from the three companies I'm going to show you guys. In any organism, including human cells, with efficiency and precision. The power and versatility of CRISPR-Cas9 has opened up new and wide-ranging possibilities across biology, agriculture, and medicine. Doudna and Charpentier were recognized by the Nobel Foundation for their discovery that a gene-cutting molecule Cas9 used by bacteria to kill viruses can be re-engineered as a precise and easy-to-use gene editing tool. So Doudna was focusing on RNA, a partner to DNA, in carrying genetic information and a key part of several molecular machines, such as the ribosome and telomerase, that help DNA do its job. She became interested in CRISPR because of her long-standing interest in such RNA-based machines. Her attention was first drawn to CRISPR by a UC Berkeley colleague, Jill Banfield, who encountered it while studying bacteria living in extreme environments. The system intrigued Doudna, who wondered how exactly an unusual repeating sequence of DNA in the bacterial genome enabled bacteria to mount a successful defense against viral infections. What they and other researchers pieced together is that when viruses invade bacteria, the bacteria cut up the viral DNA and place these pieces in their genome, like most wanted posters. So it gives them immunity. The viruses and bacteria have been having this battle for millennia, right? And this is how bacteria have CRISPR, humans don't. We're taking this technology from bacteria and we're utilizing it in human genomes. Charpentier was focused on the type 2 system CRISPR-Cas9, and in a paper published in Nature in 2011 described an unusual RNA not found in other CRISPR systems, the tracer RNA, and how it works together with the Cas9 protein to help generate the RNA copies of the most wanted posters. She and Doudna then considered how the Cas9 protein might also be involved in the final step of CRISPR immunity, surveying the cell and cutting up invading DNA. The two began their collaboration the year after meeting at a scientific conference in Puerto Rico. A year later, in the 2012 Science paper, Doudna and Charpentier rocked the scientific community by showing that the protein Cas9 is guided by both the tracer RNA and an RNA matching a viral sequence, which it uses to seek out and destroy matching viral DNA. The two scientists also demonstrated that this cellular defense system had applications beyond killing viruses. They engineered the two-piece RNA into a single RNA and showed that it could be designed to pinpoint any gene in any species, not just bacteria, allowing the Cas9 protein to cleave just that spot. Doudna and Charpentier then made the proposal that CRISPR-Cas9 can be repurposed to be an exceptionally powerful tool for editing plant or animal genomes, including human genes, and customized to delete or add specific strands of DNA. She actually founded two companies. One is Caribou, and the other one is Mammoth Biosciences. So Mammoth Biosciences is a San Francisco-based bioengineering tech startup. The business is focused on improving access to biosensing tests, which address challenges across healthcare, agriculture. So this is a diagnostics company. And then she also was a co-founder of Caribou, which has since been bought out by Intellia Pharmaceuticals. So we actually need to cover both of those. Mammoth Biosciences at the time of this video is not a publicly traded company. Emmanuelle Charpentier, she worked with Doudna. She's the co-winner of the Nobel Prize. She's best known for her Nobel work in deciphering the molecular mechanisms of CRISPR-Cas9 and repurposing it into a tool for genome editing. In particular, she uncovered a novel mechanism for the maturation of a non-coding RNA, which is pivotal in the function of CRISPR-Cas9. Specifically, Charpentier demonstrated that a small RNA called tracer RNA is essential for the maturation of CRISPR RNA. Charpentier met Doudna in 2011 and began collaboration. However, in 2013, Charpentier co-founded CRISPR Therapeutics. So we talked about Antelia, which Jennifer Doudna, she founded Caribou, which then got acquired by Antelia. And we talk about Emmanuelle Charpentier. She's the one who co-founded CRISPR Therapeutics, which we got to do an entire video just on that by itself. And then there's another guy who was kind of racing against both Doudna and Charpentier. His name is Feng Zhang, and he's one of the co-founders of Editas, which is stock symbol EDIT. Zhang began to work to harness and optimize CRISPR system to work in human cells. While Zhang's group was optimizing the Cas9 system in human cells, the collaborating groups of Charpentier and Doudna described a chimeric RNA design, which is capable of facilitating cleavage of free-floating DNA using purified Cas9 protein and a synthetic guide. Zhang's group compared their RNA expression approach with a design based on the Doudna Charpentier chimeric RNA for use in human cells and established features of the guide necessary for Cas9 to function effectively in mammalian cells, which are dispensable in biochemical assays. Basically, Zhang, he's a co-founder of Editas. Editas Medicine is a clinical stage biotech company which is developing therapies based on CRISPR-Cas9 gene editing technology. It was founded in 2013 with funding from Third Rock Ventures, Polaris, and flagship and licensed CRISPR patents from the Broad Institute's Feng Zhang, patents from Harvard's David Liu and George Church, and patents from Partners Healthcare. This is probably the second biggest. If CRISPR Therapeutics is 8.6 billion, Editas 2.7 billion, and then Intellia is 4.8 billion. So these are like the three main publicly traded CRISPR companies, like the three leading CRISPR companies. But if you're curious about learning more about this, I think it's interesting. I think it's worth looking into. I just wanted to give like a very brief overview of the key players, namely Doudna, but also Charpentier, and then Feng Zhang. I think it's definitely worth looking into. I think the book, The Code Breaker, as well as A Crack in Creation, which was actually written by Jennifer Doudna, are must reads. And like I said, stay tuned for my future video on explaining. I'll do my best to try to explain in layman's terms CRISPR like in under 10 minutes so you can maybe understand it. But these are the companies to look at. This is a technology that's already in clinical trials. I believe the furthest ahead is CRISPR Therapeutics, the one co-founded by Emmanuelle Charpentier. And I think they're in a phase 2 or 3 for sickle cell. And they're currently, the company has several drugs in development. These include the drug for the treatment of rare blood disorder beta thalassemia and sickle cell, which is being co-developed with Vertex Pharmaceuticals. It received orphan drug designation from the FDA. So this is the furthest ahead is CRISPR Therapeutics. It's also why they're probably the biggest market cap. But all three are worth looking into. This topic is worth looking into. I'm going to unpack it more. This is just an overview of the key players and the key companies in CRISPR. Hopefully you learned a thing or two. Hopefully you liked it. I'll catch you later, Guru Nation. Take care.